And without further ado, I'd like to introduce our first speaker, Dr. Sabri Rial from the University of Montreal. And today he'll be talking to us about the scaffold protein 1433 zeta and its interactome are required for expression of key adipogenic genes during the early stages of adipocyte differentiation. So I'm just going to get Sabri to share his screen now.
Good morning, everybody. Can you see my screen?
Yep, looks good.
Thank you, Cathy. Thank you, Matthew. Thank you, CSMB, for this amazing opportunity. It's a real pleasure for me today to present a summary of my postdoctoral project conducted here in Montreal in the laboratory of Professor Gareth Lim for the Centre de Recherche of the CHUM belonging to the University of Montreal. Me, as a specialist, as a scientist, I'm really interested in understanding and deciphering the mechanisms that could trigger, that are somehow associated to obesity. Because, excuse me, trying to move forward. Exactly. Because obesity presents a global issue worldwide. I think we all know that the prevalence of obesity is highly alarming, with more than 600 million persons affected by obesity and more than 1.1 billion individuals at least overweight. It's well known that obesity consists in an excessive accumulation of fat in a body, mainly driven by a high abundant availability of energy-dense nutrients. And the reason why obesity is a problem is also because of its relation with other chronic disease, such as insulin resistance and type 2 diabetes, metabolic disorder associated steatotic fatty liver disease, and also cardiovascular diseases. Of course, if I start to talk about the obesity burden, I can maybe take the whole session. It's not necessarily the purpose, but you can, of course, read more about the obesity burden via the CDC information. But lastly, and not the least, obesity represents an enormous burden in terms of cost for public health, which is estimated to exceed $4 trillion in the world. To better understand obesity, it's important to, of course, decipher what is underlying to obesity. And among the mechanisms underlying obesity, there is the physiological process of adipogenesis. Adipogenesis is the biological process that consists in the differentiation of adipocyte precursor cells into mature adipocytes that in turn could, in the obesogenic context, expand both in size and number. And the natural purpose, the function of adipose tissue is to store excess energy into fat, but of course, such expansion could trigger obesity and related diseases. The molecular mechanisms that underlie adipogenesis are quite complex, but they are somehow well studied. The differentiation from APC to adipocyte is supported by molecular pathways that, in brief here, I will not describe everything in detail, but in brief, they consist in the early activation of key adipogenic transcription factors, such as CABPs, and also a late one called PPAR gamma, the master upregulators of expression of adipogenic genes. How sorry, I'm trying to hide this. Okay. How these molecular pathways are well studied, little is known about the molecular factors that tightly coordinate these molecular pathways in space and in time. For this reason, in the lab, we are interested in studying the 1433 protein family. These proteins are molecular scaffolds, and their function is to tightly coordinate signaling and molecular pathways in both space and time. They are highly conserved among species, and in mammals, including humans, of course, they consist in 7 mammalian isoforms. They are ubiquitous, found in all cells and everywhere in cells, and their mechanism of action is pretty simple. It consists in PPI, so peptide-peptide interactions. And 14-3 protein can recognize specific phosphoserine and phosphothreonine motif on a broad variety of client proteins, then interacting with them and regulating them. And through regulating this broad variety of proteins, 14-3 family is able to coordinate, of course, many cellular pathways, including cell fate decisions. Then, more exactly, my mentor, Gartlim, in his pioneering work previously, he established that 14-3 zeta isoform specifically is required for adipogenesis, as its depletion in 3T3L1 pre-adipocytes completely blocked the potential of the cell line toward adipogenesis, as imaged here by oil redo staining and quantification of oil redo staining accumulation. Further, he saw, sorry, what is that? Okay. He further saw that whole body knockout of 14-3 zeta in mice significantly delayed body weight gain, significantly also delayed the formation of visceral white adipose tissue, and significantly reduced also the size of white adipocytes. He then established, using the 14-3 zeta whole body KO, that this isoform allows adipogenesis through regulating cell cycle of pre-adipocytes, and also through protecting CBP adipogenic transcription factor against autophagy. Then, when I joined the lab, we noted the major limitation of, we discussed the major limitation of a whole body KO is that it makes us unable to distinguish the individual contributions of specific cell types. Hence, we came up with our question: does 14-3 zeta exert its regulatory function in adipose-specific cells? To address this question, using the famous Krelox system, we generated a major adipocyte-specific deletion of 14-3 zeta in mice to assess its impact on adipogenesis. And quite surprisingly, we saw in males that major adipocyte-specific depletion of 14-3 zeta did not impact body weight progression, nor did it impact adiposity. This was the case in males, but it was also the case in females. So, of course, at first it was somehow disappointing, but we pushed the question a bit further, and we just moved the cursor toward the adipose precursor cells that also represent a population of adipose tissue, and we assessed this time if the adipose precursor cells, so APC specific depletion, deletion of 14-3 zeta could trigger any effects. And interestingly, we saw that in males, the APC specific deletion of 14-3 zeta significantly lowered body weight gain, and either event was not significant, but tended to reduce fat mass accumulation in males. While in females, quite surprisingly, we had an exact opposite phenotype with HPC-specific deletion of 14-3 zeta, significantly increased body weight, and significantly doubled the adiposity as shown here by fat mass measurement, by also the white inguinal and perigonadal adipocyte sizes. Then taken together, these results suggested that in adipose tissue, 14-2-3 zeta exerts its adipogenic regulatory functions in adipocyte precursor cells and not in mature adipocytes. Hence, our hypothesis that our initial hypothesis that 14-2-3 zeta enables adipogenesis through recognizing, binding, and then nucleating key adipogenic transcription factors during the earliest steps of adipocyte differentiation belonging to APCs. To assess this, we decided to interrogate the changes of the nuclear interactome of 14-2-3 zeta during early steps of adipogenesis. To perform this, we generated, in collaboration with Professor Thomas Durkan from McGill, our TAP33L1 model. It consists in 33L1 cells modified using CRISPR-Cas9 in such a way that they express a TAP-tagged version of 14-2-3 zeta. And such a TAP tag was supposed to allow specific purification of 14-2-3 zeta to decipher its interactome. We further confirmed that the TAP33L1 cells, as compared with the wild type or the non-edited 33L1 cells, conserved its adipogenic potential, or in other words, conserved its responsiveness to MDI and MDIR differentiating treatments, as imaged here by already staining, but also as quantified here by already staining, triglyceride accumulation, and lipolytic potential, as well as expression and nuclear translocation of PPAR gamma, again, which is the master upregulator of adipogenesis. Once we confirmed that our TAP33L1 model was valid in terms of adipogenesis, we submitted it to early time points of differentiation with 24 and 48 hours of MDI treatments before isolation of nuclei and purification of the nuclear for TAP 14-2-3 zeta complexes in view of trypsin digest, and the purpose was to interrogate the interactome changes of 14-2-3 zeta. And we saw, interestingly, that at 24 and 48 hours of differentiation, respectively, the 14-2-3 zeta and the nuclear interactome was enriched with 62 and 113 peptides. But almost none of them were transcription factors, surprisingly. They were either, these interactors were either categorized by gene ontology as associated to chromatin modification functions. We effectively saw that with our proteomic, 14-2-3 zeta physically interacts with chromatin modifying enzymes such as CBX5, also DNA methyltransferase family, and also histone deacetylase. Of course, physical interaction doesn't necessarily say something in terms of function. We then, for this reason, we also confirmed functional relation between 14-2-3 zeta and these chromatin modifying enzymes. You can see here in 2-2-L1 cells that silencing YYZ, the gene that codes for 14-2-3 zeta, significantly reduced the activity of DNA methyltransferase. But also, interestingly, we can see that during differentiation of non-transfected cells, the early time points of differentiation are associated with increased activity of histone deacetylase, which is not anymore the case when we deplete the cells from 14-2-3 zeta. And in the opposite way, we see that the early differentiating steps are not associating with changes in histone acetyltransferase activity, unless when we deplete 14-2-3 zeta. In other words, taken together, these results indicate that 14-2-3 zeta during early steps of adipogenesis balances the activity of chromatin modifying enzymes. Okay, but does it result effectively in any modification in terms of chromatin accessibility. Well, of course, as we came up with this question, we revised our initial hypothesis, and we either proposed that 14-2-3 zeta redefines the accessible chromatin landscape of APCs during early critical stages of adipogenesis, likely through, of course, interacting and regulating chromatin modifying enzymes. To question this hypothesis, we performed attack sequencing on 33L1 cells treated or not with Si14-3 zeta and submitted, of course, or not to early time points of adipogenesis. After amplifying and sequencing the differentially accessible chromatin region in collaboration with Dr. Aida Martinez-Sanchez from Imperial College, UK, we found, very interestingly, that depletion of 14-3 zeta results in the reduced accessibility of several hundred genes during early steps of adipogenesis. And in the same time, it results in the aberrant increase of several hundred other genes. In other words, effectively, deletion of 14-3 zeta at early steps of adipogenesis redefine the accessibility, the accessible landscape of chromatin. Among, the genes that were reduced in terms of accessibility at 24 hours were mainly associated to development, while the genes that were reduced in terms of accessibility at 48 hours time point were mainly associated to function of accumulation and anabolism of esterified lipids. In other words, the function that we lose that we potentially lose 24 and 48 hours due to the closure of chromatin accessibility after the loss of 14-3 zeta correspond to adipogenic signature. As an example, we saw that fatty acid binding protein 4 and adipogenin are two key factors involved in adipogenesis are decrease in terms of accessibility after deletion of 14-3 zeta. But also among the enhancers and the promoters that were downregulated in terms of accessibility, we found the peroxisome proliferator activating receptor response element, the response element of PPR gamma, but also PPR alpha and also retinoic X receptors. And these regions are absolutely crucial for differentiation of any, of course, adipocyte. And again, they are lost when 14-3 zeta is lost. Okay. But does these changes in accessibility really result in changes in expression of the corresponding genes? To question this, we overlapped our attack sequencing data with the previously generated RNA-seq data, previously generated from cells treated exactly with the same manner. And overlapping the data showed that at 48 hours, not at 24 hours, but at 48 hours, we have a positive correlation between the accessibility of the promoters and the expression of the corresponding genes. And more exactly, we defined here that when we deplete cells from 14-3 zeta, we identified 3 genes that were downregulated in terms of promoter accessibility and RNA expression. It was fatty acid binding protein 4 again, but also resistin and FAM83A. FAM83A and resistin has been previously established as critical for the progression of early steps of adipogenesis. Then, to have a kind of double confirmation, if 14-3 zeta is involved in chromatin accessibility, we assessed if the overexpression of adipogenic transcription factors could rescue the effects of 14-3 zeta deletion. For this, we used the NIH33 cells that are usually unable to differentiate into adipocyte. They are unable unless if they overexpress these canonical adipogenic transcription factors. You can see here that pushing these ATF toward overexpression, followed by MDIR treatments, could indeed increase the expression of adipogenic. But such an increase is significantly lost when NIH-33 cells are previously treated with SI-YOZ. In other words, we can push we can overexpress adipogenic transcription factors as much as we want, but as long as 14-2 zeta is missing, we cannot push to expression of adipogenic genes, meaning that 14-2 zeta acts way downstream of the canonical pathway, and downstream of this pathway is the chromatin. As a graphical summary of this finding, we propose here that 14-3 zeta at the early stages of IPC's differentiation toward adipogenesis may interact with and regulate, either directly or indirectly, the activity of chromatin modifying enzymes in such a way that chromatin regions corresponding to adipogenic genes are accessible, and in such a way that adipogenesis could be triggered. And this new mechanism is to put in maybe in parallel to the previous mechanism defined by the pioneering study of Garrett a few years ago. Our findings are found here in our bioarchive that has been uploaded last week, and please, you can feel free to read it and to come up with any criticism or question. It's time for us to integrate these kind of discussions, of course. Lastly, I want to thank, so many persons and institutions, but I need to be brief. So I will warmly thank, of course, Montour Garrett Lim, but also the laboratory that you can see here, our collaborators. I need to acknowledge the precious importance of our collaborators during this work, the facilities of the CRHUM that are absolutely amazing and helpful. And of course, I want to thank our funding agencies and the CSMB series for having me today, allowing me to present my work, and of course, the audience for listening.
All right. Thank you so much, Sabri. That was great. So if anyone has any questions, you're welcome to either raise your hand or drop them in the chat. And I'll just give everyone a minute. Go ahead, Matt.
That was a really nice talk. Really nice work. That's awesome. I was very interested in the sexual dimorphism that you're seeing in the mouse model. So I guess my question is kind of two parts. Can you speculate on like why that might be? And does that have any impact on your cell models? Like what's the XY status of the cells you're using?
Dear Matt, these are excellent questions. And the questions that we're asking actually since the beginning, and we noted this sexual dimorphism in specifically in the 14-3 in the IPC specific depletion of 14-3, and to be straightforward, what we suggest as a possible underlying mechanism is the estrogen receptor signaling versus the androgen receptor signaling too, because it has been clearly established mechanistically speaking in other cell lines, mainly breast cancer cell lines, a close and physical interaction between 14-3 zeta and also the estrogen receptor estrogen complex. And such an interaction was shown to regulate the localization, the intracellular localization of this complex. So we we think that there is something at play here with sexual hormone determined adipogenesis, because as we when we compare, for example, here the fat mass of adipose specifically, APC specific depletion of 14-3 zeta, we here reach levels that could somehow correspond to wild type males. Okay, so are we losing by doing such deletion what characterizes the genoid adiposity? This is something that we want to assess and to yeah, to be transparent, we were such interested that we submitted an application for a grant to review this, and we're still waiting for the result of such grants. So we hope that we can come up with a better more detailed answer for that. As for the cell model, 33L1 cells are males. Okay, so those who are asking, yeah, they are males. And of course, the study is maybe somehow biased by the fact that 33L1s that we use for the molecular characterization are males. And I think, yeah, it would be interesting for further studies to come up with maybe stem cells, I mean, stemming from males and females, to see if we can repercute these differences in vitro.
Cool. Yeah, thank you. That's another great example of how you need to study both male and female in your models.
Yes.
It's really nice.
Exactly. The fact that we included the males and females since the beginning actually pushed us to find this dimorphism and to ask for another grant to study this fantastic issue, and hopefully we will be able to study it. Exactly.
Awesome. Feel free to connect with Sabri if you guys have any more questions. For now, we're going to switch over to our next speaker. So I'd like to introduce Dr. Zoe Gillespie from the University of Toronto, who's going to be speaking about epigenetic changes are distinct between differing methods of labor induction in the mouse. So take it away, Zoe.
Great. Hi there. Thank you for having me. Well, I'm just sharing my desktop screen right now. Let's try the correct share screen. Here we go. Hopefully you can now see my PowerPoint slide. So of course, like Kathy introduced me, and thank you for introducing me. My name is Zoe, and I'm a postdoctoral fellow at the University of Toronto here in the lab of Jennifer Mitchell. And one of the things that we study in this lab is how we can regulate or how labor itself is regulated. So before I get into the exact epigenetic details too much, I wanted to talk a little bit about what preterm labor actually is, because of course for us to so basically preterm labor is when a baby is born anytime between this 37-week time point. And unfortunately, by being the earlier a baby is born, the more likely that child is to have a decreased survival rate. And not only that, but they're more likely to have early life and late life morbidities. So including things such as underdeveloped lungs in early life, and things such as cerebral palsy and neurodevelopmental disorders later on in life. What exactly causes labor, especially at the genomic level, is pretty poorly understood. We know in term labor, there's some sort of functional hormone withdrawal in humans and an increase in tissue contractility, whilst in preterm, this is often associated with things such as infection, trauma, stress, and even age of the mother. However, again, exactly how those result in increased tissue contractility and parturition is poorly understood. So we are interested specifically in the myometrium. So we've got a little figure of the human myometrium here on the top right. And this is the tissue that's basically responsible for that contraction during labor. It's a highly dynamic tissue. It goes from this quiescent state, and upon pregnancy has to grow massively in size. So early on in the first trimester, we're going to see the cells going from this quiescent state into this proliferative state. We know this as hyperplasia, but not only do these cells have to grow, or not only do we need more of these cells, but they also have to grow in size themselves. So they get bigger, and this is what we call the synthetic phase with hypertrophy, and also lots of extracellular matrix remodeling. Eventually, what we're going to see just in the run up to labor is development of gap junctions and expression of contractile proteins, which is going to help the tissue act as a whole when it comes time to having to push baby out. Now, the thing is, obviously, it's very difficult to collect myometrial tissue from a pregnant woman. Often this is going to have to involve taking tissue during C-section. If a woman is having an unplanned C-section and she's actively in labor, normally that's an emergency, it's very hard to get consent. It's even more difficult to get tissues at these other stages. So instead, we use this mouse model. And this mouse model is a really, really good proxy for the human myometrium. We see similar changes in terms of myometrial development. And again, we have that withdrawal of progesterone just prior to labor, so very similar laboring mechanisms. And this allows us to collect tissues at what we would consider preterm labor as well as at labor. So this is something the lab has been looking at for a little while in terms of, we've looked a little bit into this. So before we understand labor, we know that we have to, or before we understand preterm labor, we know that we have to basically understand how labor works. And because the Mitchell Lab's been working on this for a little while, we've already used this model and done some RNA-seq to look at gene expression to understand how gene transcription may change between this non-labor and laboring state. Whoops. And so what we see here, I've just lost my screen. I'm going to apologize. Can we return that? Okay, there we go. Cool. So what we saw is that day 15 to labor, day 19, which is what we consider term labor, we saw this big change in gene expression profiles. What we didn't necessarily see, though, is changes in histone modifications, which led to this idea, or at least we didn't see these changes late in labor. We saw that these were established very early on, which led us to the idea that actually the myometrial tissue is set up in advance of labor and is epigenetically regulated. Specifically, this data allowed us to identify some potential transcription factors that are associated with promoting contractility gene expression, such as gap junction alpha 1. So from this, we have these ideas of these transcription factors such as GUN and FOS. These belong to the AP1 transcription factor family. And this story is continued a bit by Nora, a PhD student in our lab, where she then went to do ATAC-seq in the same models. So ATAC-seq allows us to examine open chromatin and closed chromatin and how that's changing within the myometrium dependent on labor. And so what she saw is actually there's this very big opening of chromatin at term, but not in labor. And again, this very big opening was enriched in AP1 transcription factors such as GIN-D and FOSL2. So what we wanted to do is take this, and now that we have some sort of understanding of labor, we wanted to extend this to the idea of preterm labor. So we have two models of preterm labor in the mouse. The first model is mifepristone. So mifepristone is actually the compound that's used to induce labor early on in kind of very like first trimester labors in humans, I suppose. And this is a withdrawal of progesterone type method. So we do this in mice, and this will reliably induce labor 12 to 24 hours after induction. The other method we use is an infection-based model using lipopolysaccharide or LPS. So this is a sterile infection model, and again, 12 to 24 hours later, labor is successfully induced in these mice. And we collected the tissue of the myometrium from these mice during active labor and sent that for ATAC-seq. So the first thing I wanted to do is, as soon as we got our ATAC-seq data back, I wanted to check, hey, are we actually seeing changes that we would expect? So when we look at ATAC-seq data, the peaks are going to be representing open chromatin regions, while lack of peak indicates that that region is closed. An open chromatin region suggests that regulatory elements could, or proteins could bind within that peak and help regulate the process. So so we see here we've got a peak at the gap junction alpha-1 gene. That's, again, the gene we're interested in because it results in contraction during labor. So step one is, yes, we have a good control for seeing that actually the ATAC-seq peaks look a little bit like what we would expect them to in both response to LPS and mifepristone. So I wanted to look at this in a little bit more detail now and look at all the regions that were changing in chromatin inaccessibility in response to mifepristone or LPS. We saw that in response to mifepristone, we had a pretty even opening and closing of chromatin, about 108 open regions and 117 regions closing, including OXTR, which again is associated with labor. Yeah, so when I took these open and closing regions, I then wanted to see whether or not I could associate these with transcription factor binding motifs. And what we saw is that in the opening of chromatin in response to mifepristone, there's a huge cluster of opening and enrichment for AP1 transcription factors. So all these FOSSes, these GINs, these GIN-Bs, these FAT-F, this GIN-B, all of these we know have previously been associated with labor. So to us, it looks like mifepristone is looking very similar to term labor. We then wanted to do the same, but using LPS. So this is our infection-based model. And we were pretty surprised because not only is the magnitude of change a lot bigger, right? We're seeing 500 regions open, 2,600 regions closed. It's the fact that we've got this big closing down, which is in contrast to what we showed earlier at term labor, where we saw that opening chromatin. So this shutting down, we've then associated with genes. So we took these regions, these chromatin regions, associated them with the nearest gene and did Go ontology. You'll note that I didn't show this for the mifepristone, and that's because surprisingly, even though we could associate those regions with genes, we didn't actually get any results for the Go ontology analysis. However, with LPS, we're seeing shutting down of pathways associated with tissue morphogenesis and development, especially with this mesenchymal epithelial transition. When we look at what's opening, this is a little bit more what we would expect, because what we're seeing here is we're seeing upregulation of immune system processes, we're seeing cellular response to molecular bacterial origin, inflammatory processes, right? And we already know that labor is somewhat inflammatory. We know that those factors have been associated with labor previously. And of course, we've created, this is an infection-based model. So we'd expect some of that to be happening. When we then did some footprinting analysis here to see whether or not, to see what transcription factor binding motifs were being gained and lost, we saw that many transcription factor binding sites, such as NFKB1, REL-A, again, were associated with inflammation. Whilst in the lost chromatin, we saw loss of SOX factors, PAX6, PAX2. What's pretty cool here is we're actually seeing families of transcription factors come up, which means that we've got a pretty good indicator of which motif specifically we need to be looking at and what's going to be changing in future studies. I wanted to assess how common LPS and mifepristone, or how alike mifepristone and LPS are. And I was actually pretty surprised by the limited overlap between these two models, right? These are both preterm labor models. I thought we would identify a family of transcription factors that were in common, and then we'd have a nice set of targets for preterm labor and how to stop labor. Naturally, that's not exactly what happened, and I actually only saw 3 binding motifs overlap between LPS and mifepristone. But interestingly, we saw a much greater number of overlap between the closed chromatin or lost chromatin regions. Now, what I then wanted to kind of link onto is the idea of these factors, how those linked back to labor, and how much regulation there was in common between mifepristone LPS and term labor. So referencing back to the data our lab has previously produced in the mouse myometrium, I crossed this all over, and again was very surprised that then between all three conditions, mifepristone LPS and term labor, I only had one transcription factor binding motif shared between the three. So of course this is going to be a target of interest in understanding how this regulates the laboring process, and also potentially how we could use this factor to prevent preterm labor. I also noticed a lot of overlap between mifepristone and term labor, but not so much common between LPS. When I looked in further detail at these 56 common transcription factor binding sites, we saw lots of SOX factors come up, as well as this estrogen receptor 1. This is really interesting because, again, Nora in our lab has previously shown that done work on SOX and the regulatory role it may have in term labor, specifically conducting luciferase assays looking at gap junction alpha 1, our contractility gene, and as well FOS that we previously knew was involved. And we saw that depending on the context, if SOX is introduced to the regulation of gap junction alpha 1 promoter with GIN and D, there's that increased reporter expression. The same here with FOS. So this suggests that SOX4 and SOX factors may be involved in some sort of regulatory switch in labor between maybe quiescent and laboring myometrium. I then wanted to pay attention to these other 5 transcription factors that were overlapping between all 3 conditions. And I had a quick check to see what was expressed in our tissues. And so we found that this HNF1A, B, and ARID3B were common and expressed in our myometrial tissue. LHX4 wasn't expressed, but probably came up because its transcription factor motif is so similar to that of ARID3B and HNF1 alpha. So this has led me to conclude that we've got a couple of potential things going on. First of all, we can say that actually term labor and mifepristone are quite similar to one another mechanistically. It's likely we've got these GIN and FOS factors involved in regulating expression of contractility associated genes, and that SOX is in there acting as some sort of switch. What's especially interesting is, despite being another labor and preterm labor model, it looks like LPS is doing something different to term labor and mifepristone at the genome level. So looking at these cis-regulatory elements, it looks like we've got somehow REL-A and FKB1, these inflammation-associated transcription factors, again, regulating expression of gap junction alpha 1. And these binding sites are present upstream of this gene. So 2 independent mechanisms regulating that can actually lead to the same outcome, labor. And then we've got this ETV6 in common. We want to know, how is this joining this regulatory process is going to be a big question. And finally, we noticed that actually maybe because we've got more overlap in what is lost during during labor, perhaps these usually are involved in maintaining some sort of myometrial quiescence. And maybe it's maintenance of myometrial quiescence or non-laboring state that we need to be considering when it comes to some of our next preterm labor druggable targets. With that, I want to thank everyone in the Mitchell Lab, especially Nora, who's contributed a lot to laying down the work ahead of this experiment, as well as Valana Shuka, who's now a postdoc. And also our collaborators at the Sinai Health Region and Balloon Tannenbaum Research Institute and our funders at CHR. And I'm happy to take any questions.
All right. Thank you so much, Zoe, for that great talk. We're going to open up the floor now for any questions. So feel free to either type them out in the chat and I can read them out, or raise your hand and we can unmute you to ask your questions. Go ahead, Sabri.
Yes, can you hear me? I always struggle
Yeah.
with the microphone. Oh, okay, thank you. Yeah, it's a fantastic talk, dear colleague. I was wondering if there was this factor in common that you found with your ataxic. It's ETV6 or something.
ETV6. Yeah.
ETV6, yeah. I was wondering if you were planning, Zoe, to go maybe further in generating maybe any KO model of this this factor, and to see if you compromise your baby yield or something with the mice.
So definitely, I think that it's a target we're interested in. The motif itself actually is very similar to some other targets that we've identified previously. And of course, we know transcription factors and this predictive idea of footprinting. If the motif is similar, there's a good chance that it's kind of somehow it's kind of fair game for that knockout, right? So we are definitely interested in doing some knockout models. Our lab has a lot of experience with CRISPR-Cas9 and so I'm currently working on a cell model version of this process, but also perhaps we'll be doing this in the mice themselves to see whether or not we can, if changes the labor timeline.
Oh, interesting. And do you know if any, I don't know, maybe if it's really applied to this field, because I often think metabolically, but I don't know if this field exactly, if there are any association studies, genome association studies that are done that can link this gene to any condition of endometriosis or any kind of issue relative to uterus.
I actually haven't looked specifically to see if there's any GWAS studies for the ETBS and whether it's linked to endometriosis or anything. I know there are a lot of researchers doing work like that and looking at potential interventions and how that's regulated. But yeah, I don't know that one, I'm afraid.
Alrighty, we have one more from Jeffrey. So great talk. Do you know if human preterm labor is commonly associated with inflammatory conditions?
So that's a really good question. I know that it's associated with infection. Often infection does lead to induction of preterm labor, and that's why we have that LPS model. Human inflammatory conditions is something I'd have to go do a bit of a literature dive for, but it would again be very interesting to see. Actually, I wouldn't be surprised to see some association of preterm labor if the mum has a disease that's inflammatory or some sort of flare up with seeing preterm labor as a result.
If there are no more questions, then I'd like to give our speakers another round of applause for the great talks we saw today. And I'd also like to remind everyone that next month we will be meeting again for our May installment of the CSMB seminar. So feel free to join us and have a great rest of your day. Bye-bye.